All right, hello everyone. This is the Silently live stream for Heredity B. And so I'm just going to go like a brief overview of this event, go through the rules, just some basic tips. So, if you haven't already, Heredity is an event about genetics and some of the molecular biology. And yeah, um, these are some of the topics. They'll be listed in the Silently B manual. Make sure you have the right year, 21. And this is a little bit outdated. Yep, make sure you read the rules, read over. But the Solid Wiki is a great start to learning some of the material. And essentially, you want to like go further in, just like watch more videos on YouTube or read a textbook. Always a great idea. You can always make Quizlets to make sure you remember all these, because a lot of this is a lot of information, and kind of like your cheat sheet, like a good basis for your cheat sheet. Take a lot of practice tests. They are on silently.org. Yep. Split the tests. Yep. And just stay calm and focused. You should be good. All right. So I'm going to go right into the content. And so first off is just what are genes and proteins? So genes are just segments of DNA that help encode for proteins. So if you think about it this way, it's kind of like a chart where you have DNA, which encodes for mRNA, which encodes for tRNA, or which brings tRNA, which brings an amino acid, and eventually your amino acids form a peptide chain. You ultimately get protein. So the two main processes that are involved with the production of a protein are transcription and translation, and I'll go further into that next. And so basically, you have your DNA in your cell, and you want to get the mRNA from that. Basically, getting like it acts as like a template, and you get your information from that. With the production of mRNA, it travels to your ribosome, or originally this process occurs in the nucleus, the transcription. And so mRNA will be transferred to the outside of the nucleus into the ribosome, as you see here. And there are three sites: the elongation site, the propagation site, and the German site, or I think it's just EPA. You can look up the actual names, but they're not listed here. So, but in a sense, your mRNA will serve as a template for your tRNA, which will bring you the anticodon. But what's most important is the tRNA brings an amino acid, or has an amino acid attached to the end of it. And so they will form bonds with each other, chemical form. And so it's kind of like an additive process where one amino acid is encoded for, and then it adds on until you hit a stop codon. And codon is just three nucleotide pair and how it works. There is a stop and a start and a stop. But as you learn more about this process, you'll learn about the intricacies and how it actually works. And so just a general overview of the DNA structure, which is kind of essential just for replication, is that you have a phosphate group, a ribose, a 5-ring ribose sugar, and your nucleotide. And so you have 4 types of nucleotides for DNA, and you have another nucleotide for RNA. And the nitrogenous bases is these 4, and the one for RNA is uracil, U. It's also very important to note that the carbons on the sugar are numbered accordingly. So this is number 1, the number 1 carbon, 2, 3, 4, 5, and DNA is synthesized in a 5-to-3 manner. It's very important to know, and I highly suggest that you look up the reason behind it and the mechanism and why it has to work that way. Because it's also very important when you understand DNA replication. So DNA is synthesized in a 5' to 3' direction. And so when you're replicating DNA, you first have this enzyme called I can't even say it. But this is right here, and it unwinds. So I go back here. You can see how it is in this double helix structure. It has to unwind it before it can actually replicate any of the strands. So this enzyme unwinds it, and the helicase will kind of unzip it, and so you get this. You get two separate strands, and you will have a leading strand which will work in a 5-to-3 direction. It's continuous. There's no interruptions because it is working in the way that it's intended to. But the lagging strand does not, and so you will have to use RNA primers. And primers are basically just matching stuff, or the match to the base pair in which is in the original DNA. And so it'll have to work in a 5-to-3 direction. And so you kind of get these fragments because you're not working in a continuous manner. And you also have to use primase to kind of glue the strands, the fragments together. But yeah. And so DNA polymerase is the main protein or the main enzyme that is working. And it just kind of puts the nucleotides together. I highly suggest that you research DNA polymerase and RNA polymerase, because those two polymerases there are different types of polymerases too. And you should be familiar what their function is. And yeah. And so mutations, there are point mutations, insertions, deletions, duplications, and inversions. Although the primary focus of genetics or heredity is going to be what are the effects of point mutations, insertion, deletions, and duplications. And so your silent mutations are when something changes, or okay, I'll start off with a point mutation. It's just when one nucleotide is changed at the DNA level, which kind of affects like your mRNA, which affects your anticodon, and ultimately affects like what kind of amino acid. So one type is silent, and so your silent mutation is it doesn't affect the protein. It's just a different nucleotide, but it doesn't affect like your ultimate end goal product, which is your amino acid. Nonsense is when the nucleotide change induces a stop codon, which prematurely ends the chain, or yeah, prematurely ends the chain, and so you're kind of cutting off a lot of the amino acids. Missense is when it changes the protein or changes the amino acid, and so silent is when it doesn't, and missense is when it does. And insertion deletions are very, very bad because they shift the reading frame of your amino acids and can completely change all of your amino acids if you're unlucky. But yeah, inversion is when things are swapped around. Duplication is when something is repeated again and again. So CT is repeated twice. But yeah. And then the next process is mitosis. But first, we should go over the cell cycle. And the cell cycle ultimately has 4 phases: G1 phase, the S phase, G2 phase, and mitosis. And so the G2S or G1S and G2 are all part of the interphase, and they are basically prep for the mitosis. And so if we start from G1, G1 is just duplicating your cellular contents, your organelles, making sure you have all your products inside. There are, if you can imagine, checkpoints in between every phase that make sure that everything is intact, proper. If not, they will go back to a G0 phase, which reads cell cycle arrest, and can undergo apoptosis or can be fixed. And so your S phase is when your chromosomes are duplicated. And so you normally have an N set, but now you have a 2N set. And so, yep, it duplicates the amount of genetic material. And G2 just makes sure everything's fine. I highly suggest you watch videos on like the actual process. And once we're after the G2, we are into the mitosis phase, and this is where the real process begins, although it's very short. And so first you have, or so normally in interphase, you have all your chromosomes. They're very loose, not very condensed. So in the prophase, your nuclear envelope kind of like dissolves, the chromosomes start to condense, and your spindles start to form. And so in the metaphase, you will have all the chromosomes line up on a metaphase plate, hence, metaphase. And then you have your anaphase, which the chromosomes are pulled apart. And then telophase, where the two nuclei start to form around the two separated chromosomes. And so at the end, you have another process called cytokinesis. It's not in this picture, but a cleavage, or the cell starts to get cleaved, and you'll form two daughter cells, which has genetic, or two cells that are genetically identical to each other. And so next process is meiosis. And so this is sexual reproduction. The other mitosis was asexual mitosis. It happens in somatic cells, but these happen in germline cells. And so essentially you're having the same process with prophase, metaphase, anaphase, and telophase, except for it occurs twice. However, what is different is that you have recombination occurring, and chromosomes can be swapped over, and you don't get homologous chromosomes, or you have homologous chromosomes, but you don't have the same chromosomes as you started with, because they could or could not undergo recombination. And so you see how when they line up during prophase and metaphase, or during prophase, is when synapsis occurs, and so they line up together, and ultimately it can be swapped. Metaphase, the same stuff, and it'll line up, get pulled apart, and then two new cells will be formed. Prophase, you have the same process again. They'll condense, spindles will be formed, another pulling apart, and what's different is that you produce 4 gametes, or that are haploid, and so they only have one set of chromosomes, and they're very genetically different to each other. And so sex determination is just taking what's it called? These two taking the gametes that are formed and undergoing fusion to form a zygote. And so what you need to know is that 2 X chromosomes are associated with female, and 1 X chromosome and 1 Y chromosome are associated with males. Genotype versus phenotype. So genetic information ultimately encodes for physical traits plus protein. If you remember from before, genes encode for proteins, or genes are the basis, kind of the blueprint for proteins. And proteins are often associated with a physical trait, because sometimes your genes can code for enzymes. These enzymes are associated with the physical trait that they help produce. And so genotype, in a sense, would be, actually, it's more important to go over an allele. An allele is a gene variant. And so you can have dominant alleles in which their expressions are dominant to the recessive and will be expressed if they are there. However, recessive, both alleles need to be recessive before characteristics can be expressed. Some basic terms: homozygous, or 2 identical alleles, so GG, both uppercase and both lowercase, are homozygous, and heterozygous are when they have both. So 2 different alleles, so the uppercase G, or the dominant G, and the recessive G. And so this is just an example of what a cross would look like. And so from before, just to nail it, the coffin in, genotype is your genetic information. And so it is the alleles are the genotype. And your phenotype is what actually occurs. So because you have a heterozygous, dominant and recessive, you produce a green pod, and that is your phenotype, the green pod. Whereas your uppercase G and lowercase g are your genotype. And so monohybrid crosses are taking these gametes, and so imagine on a gene there's an allele lowercase g, that is your allele, and so um, on a chromosome there are two alleles, and so they independently insort, and as mentioned before, gametes can form with other gametes, and that is the allele that they carry in each gamete is what ultimately produces your phenotype in your offspring. So for monohybrid crosses, first you would identify the genotype of both parents and put that on your Punnett square. And you would write each gamete in each square outside, and then you add the gametes together, cross them together. And so dihybrid crosses are just between two different genes. It is for two traits, could be for two traits. But the main takeaway is that there's two different genes now. So instead of just having the G gene, you'll also include like a R gene and a Y gene. So it's more complicated. So first, you want to determine what kind of gametes your parents are going to produce. So you undergo this FOIL process where you distribute the first gamete with the first gene with the second gene, and then you take your second allele of your first gene and distribute it again. And so you want to get rid of duplicates and use just whatever unique ones you have. So you repeat the steps with the monohybrid cross once you have identified your gametes. And then you use the FOIL process, and then just kind of do the same process for monohybrid crosses where you cross each gamete. Heterozygous mating will produce a phenotypic ratio of 9:3:1 is an important ratio. That should be noted. Sex-linked traits, and so what I've been talking about before are not sex-linked traits, but they're autosomal or somatic linked, and so they're just on your regular like 1, 2, 3, 4 to 23 chromosomes. These affect the X and the Y chromosome. And so they're similar to monohybrid crosses, except for they're on your sex chromosomes, which is the X or the Y. And so in the same sense, it's pretty much the same thing. You have the dominant and recessive alleles. However, what's different is that males only have XY and females have XX. So when they're, for the mode of inheritance, males are going to be inheriting or passing their Y chromosome to only male offsprings. So they'll pass their Y chromosome to male offspring, and the female will pass down their X chromosome. So in a sense, if a female has an affected trait and you produce a boy, if the female is affected with a recessive trait, your offspring, the male offspring, will automatically be affected, even if your male had the dominant allele on their X, just because of how they pass down their Y, and the offspring inherited the Y from the father, and inherited the recessive allele from the recessive X allele from their mother. Some traits can have multiple alleles, so just instead of they could have multiple alleles code for one trait and produce multiple phenotypes. This will ultimately lead in having more phenotypic classes. So normally you have two phenotypic classes with one gene, so you will have your dominant and your recessive, but multiple alleles can create many scenarios. Just an example is blood type. A gene, the I gene produces, actually I'm not exactly sure the specifics, but it has multiple alleles, and these alleles contain the A protein, B protein, and I, which is the recessive. And so in the end, you have 1, 2, 3, 4, 5 different phenotypes that can be expressed. And so an example of where multiple alleles is another blood type. And so codominance is when multiple alleles are dominant and both are expressed. And so an example is IA, IB are both dominant and produce the phenotype AB. So they're both expressed. Instead of having just a blood type of A or blood type of B, you have more, a combination, so AB. And they're expressed equally. Incomplete dominance is when the dominant allele doesn't completely mask the effects of the recessive allele, but instead you get a blending of the trait. And so you actually get a new phenotypic class when for heterozygotes. And so say you have big R, the dominant allele coding for red, and the recessive allele coding for white. Homozygous recessive will produce white, homozygous dominant will produce red. However, when you have your heterozygous, you have both alleles. And since they don't, the dominant allele does not mask the effect of the recessive allele, it will actually get a blending. And so you won't get like spotted like red or white as you would see in codominance, but you will see a blending of the traits. Pedigree analysis is kind of like a puzzle. You need to use logic and eliminate possible methods that are not that cannot occur, and so you need to find specific examples. My best advice for this is to just do a lot of practice problems and get familiar with the symbols, what they represent, and yep. It's also important to understand notation. So I and the Roman numeral 1 and 2 represent different generations. So the top parents will be the I generation, the first generation, and then the next will be the 2 generation that will be denoted by the Roman numerals. Same thing for karyotype analysis. It's also kind of another puzzle. You just have to notice what's missing. Are there anything? Is there anything different from what there should be? Are there extra chromosomes, missing chromosomes? Do they match with? Do your sex chromosomes match with your intended gender? And also know common disorders, so you can quickly identify. Identify what is wrong with your on this karyotype. And just for clarification, karyotypes just map out how many chromosomes are in a person and match. It's kind of like a map. And so another clarification for pedigree takes parents and their offsprings and matches and looks at specific traits that that you're looking for. For example, if we were tracking P pod colors, we would denote affected and unaffected accordingly. Or if a person has a disease, they could use it to track the disease. So if they have hemophilia, affected individual will have a filled in square, like a black square. And that helps track what your possible genotype could be based on what your parents had. And so this concludes the heredity presentation. If anyone has questions, feel free to ask.